I am the first to present some scientific projects today. I am Olga Razorenova. I reside in this building. I am at UCI for 6 years now. And my main specialty is low oxygen. So I study hypoxia. I'm going to introduce you a bunch of key terms today. And then I also study kidney cancer. And since I am in the stem cell building, I am doing some cancer stem cell work. So I'm going to talk about it as well. So I will start my lecture from introducing the key terms. And then there are a lot of key terms to go through. I will go one by one. So the first one, I'm going to talk about the blood vessels. I'm going to talk about oxygen. I'm going to talk about the normal level of oxygen and low level of oxygen. It is also called hypoxia. Then I'm going to talk about solid tumors and then the low oxygen tension in solid tumors, what it does for the tumor and how it poses problems for the treatment. Then I'm going to talk about the process of angiogenesis. And this is the process of building new blood vessels from the pre-existing ones. I will talk about kidney cancer. I am a kidney cancer biologist. And then I'm going to talk about hypoxia, specifically in kidney cancer, and also pseudohypoxia. This is when cells think that they are in hypoxia, but in fact, they have a high oxygen supply. Also, of course, I'm going to talk about cancer stem cells. I'm going to introduce a concept of synthetic lethality, which allowed us to develop therapeutics to target kidney cancer specifically and spare normal tissues. And then these are the inhibitors we found. One of them is called ROCK inhibitor. Also, I'm going to talk about statins. This sounds familiar to most of you. These are the drugs which are used to lower the cholesterol. So they also can be used apparently as anti-cancer drugs. So I will start from talking about the blood vessels. So this is the first key term we go through today. The blood vessel is a tubular structure of the circulatory system carrying blood and designed to transport nutrients and oxygen to the tissues and organs of the body. And also it takes waste and carbon dioxide and carries them away from the tissues and organs. When we talk about blood vessels, we talk about veins, arteries, and capillaries. And then today I'm going to concentrate on capillaries mostly. These are the smallest blood vessels. They are depicted here. So as you can see here, they have an opening where the red blood cells reside and all other cells of the blood system. And then also those tubes, they are formed by so-called endothelial cells. And then, as I've mentioned, oxygen and nutrients, this is what comes into tissues. And carbon dioxide and waste, this is what comes inside the blood vessel, and then it is taken away from the tissues. Also, what is not shown here on the left, but what is shown on the right. So when we form a blood vessel, we have those endothelial cells, which are forming a tube, but also there is this cell on the right. It is called pericyte. This is the second layer of the cells for the tube. And this is the cell very, very important because it stabilizes the tube. It stabilizes the blood vessel. It's going to be important when we talk about the tumor vasculature. So our next key term for today is oxygen. We need to talk about oxygenation, so we need to know what is the normal level of oxygen and what is the low level of oxygen, or so-called hypoxia. So why do we need oxygen? We breathe oxygen, we need oxygen, and we need oxygen because our tissues, they rely on oxidative phosphorylation process to generate energy. So we usually consume sugars. Those sugars, they need to be converted into the form of energy. It's called ATP. I think everybody knows what this is. This is a molecule, very high energy molecule in our body, and it is used for multiple cellular processes. So in order to generate ATP, we need oxygen. That is why we breathe oxygen. And then every tissue requires a certain level of oxygen to effectively produce this energy currency. And when there is low level of oxygen or hypoxia, this is when tissues suffer, and then they need to adjust. In order to adjust to those conditions, they need to rewire so-called cell signaling. So when do we encounter the physiologic conditions when hypoxia occurs, so low oxygen? So the first is very obvious, embryonic development. When the embryo starts developing, this is when there are no blood vessels. So there is no way to deliver oxygen to the tissues. So the level of oxygen is generally low when the embryo just starts to develop. But as the embryonic development goes forward, this is when the blood vessels are going to be formed. This is when the blood is going to be formed. And then this is when the oxygen starts to be delivered to the tissues. Another process we encounter on a regular basis is wound healing. And wound healing, this is when the blood vessels get damaged. Blood vessels get damaged, then delivery of oxygen is impaired, and then this is when also hypoxia is taking place. There are a number of pathological conditions I need to bring up: heart infarction, stroke, and cancer. And since I'm a cancer biologist, I'm going to talk mainly about cancer today. So I will talk first about the solid tumors. So solid tumors are any tumor in the body residing in some organ or tissue. The solid tumor is not the liquid tumor. The liquid tumor is the tumor of the blood, and solid tumor is everything else. So when solid tumors start growing, this is basically a little ball of cells which starts expanding due to cell divisions. And this is where the first hypoxia occurs. So if you look at the diagram, this is the capillary, the blood vessel, then cells residing next to the capillary. These are the cells getting oxygen and nutrients. But if you move further away from the capillary, this is where the oxygen tension drops. This is the hypoxic region. This is where it drops even more. So when cells proliferate, the blood vessel is going to be further and further away, and then the oxygen tension is going to drop. So another source of low oxygen tension comes from the fact that capillaries of the tumor are very different from capillaries of normal tissues. They are actually prone to collapse, and when it happens, the blood flow stops. When the blood flow stops, then cells become again hypoxic, and then this is where oxygen tension drops. So what are the consequences of this? So, and by the way, sometimes the blood vessels can reopen, and then the oxygenation can be restored. So it's a very dynamic system. This is something that does not happen in the tissues. This is very specific to tumor. So what are the consequences? There is a good consequence. The good consequence is that cells slow cell divisions in low oxygen, and if there is no oxygen, they die. So this is good for us. The tumor cells cannot grow as fast. But then there are a number of bad consequences. One of the bad consequences is called angiogenesis. I am going to revisit angiogenesis in a moment. It's the growth of blood vessels from the existing blood vessels. So cells in the hypoxic regions, they really want the vasculature to be there to deliver the oxygen. So they turn on this program to grow more blood vessels. Then those cells in hypoxic regions, they are resistant to chemo and radiation therapy. It makes sense. We usually deliver chemotherapy through the bloodstream. So the further away the cells are from the bloodstream, the less drug concentration is going to be delivered. And then finally, there is activation of hypoxia-induced molecular events. And here I am talking about hypoxia-inducible factor. That's a transcription factor I will talk about a little bit more. And then this contributes to metastasis. So I think all of us know that the primary tumor very rarely kills a patient, but then metastasis, this is something what kills the patient because cancer cells just go into different organs, and then this is where they destroy the organ. So hypoxia actually does make cells more metastatic. It stimulates cells to go somewhere else. So this is just to illustrate how it looks like in a solid tumor. So here in brown, we have blood vessels. And then around the blood vessels, those cells look healthy. They proliferate. But then beyond this circle, those cells look very crowded. They are not healthy. A lot of them are dead. And this is because the oxygen is not delivered to those cells. They lack oxygen and nutrients. We can see hypoxia. There are specific ways to detect it in solid tumors. So here we have hypoxic regions in red, and then the blood vessels are in green. So as you can see, there are black areas around the vessels because there is oxygen and nutrients there. But then all of those red areas, this is where the oxygen is limiting, where the oxygen concentration is low. So this is a typical picture of the tumor, that the oxygenation is generally low there. So the next key term is angiogenesis. I've already mentioned it is the growth of blood vessels from the existing blood vessels. So tumors do this all the time. They need to catch up on nutrients and oxygen. That is why they stimulate that process. So something to keep in mind. If you look at the normal tissue blood vessels, there are some bigger blood vessels, there are some smaller blood vessels. They also are all aligned. So there is some kind of hierarchy of the blood vessels. And then for the tumor, I think it is pretty obvious that this is the disorganized structure. So those blood vessels are all twisted. There is no hierarchy. So the picture is very different. It's called the chaotic organization of tumor-associated vasculature or tumor-associated blood vessels. Also, I talked about those tubes. They are formed by endothelial cells and also stabilized by this cell on the right called pericyte. So right here, for the capillaries of normal tissues, in green, there are endothelial cells. In red, there are pericytes. They are nicely aligned on top of the tubes. And then for tumor-associated microvessels, as you can see, those red cells are kind of peeling off. They are not tightly associated with the endothelial tubes. That is why those tubes are prone to collapse. So they are not very functional. So finally, we are going to talk about hypoxia or low oxygen as a poor survival predictor for solid tumors. So this is why we care about oxygen in solid tumors, because really, the better the oxygenation, the better the survival of the patients. So how do we measure oxygen in the tumors? So there are multiple ways to do it, and one of them is just sticking the electrode into the solid tumor. And then it's going to give us a reading. It's going to say how much oxygen is in that particular region. So if you look at those curves, so we do have here the overall survival probability. So here, 100% of patients live, and here, 0 patients live. So, and then this is time in months. So the upper curve, the oxygenation is high, so tumors are well oxygenated. And then by 70 months, we still have more than 60% of patients alive. If you look at this other curve, orange color, this is when we look again at 70 months, less than 40% of patients survive. And this is where they have low oxygenation, low oxygen in the tumor. So since we talked about the consequences of hypoxia, the drugs are not very well delivered, then also hypoxia is stimulating metastasis. It makes sense that when the tumors do not have a lot of oxygen, then patients survive less because they succumb to metastasis. So this is when we talk about cervical cancer and then a number of solid tumors, almost universally for every solid tumor, this is going to be the case. The better the tumor is oxygenated, the better is the survival. So I am also going to explain a little bit at the molecular level what is going on there. So when we have oxygen, then this transcription factor, it's called hypoxia-inducible factor, it gets those modifications in the protein molecule. It gets those OH groups. And then this is what is recognized by another protein. It is called VHL. It is also a tumor suppressor. It recognizes hypoxia-inducible factor when it is modified, and then it distracts hypoxia-inducible factor. So in a way, hypoxia-inducible factor can sense oxygen. So when there is oxygen, it is distracted. Now, when there is no oxygen, when there is hypoxia, we have no marks on hypoxia-inducible factor, and then it cannot be recognized by this VHL molecule, and then we have no distraction. Since it accumulates in the cell, it can promote cell survival in hypoxia. And then in case of cancer, it promotes cancer metastasis. So I am a kidney cancer biologist, and then in kidney cancer, of course, the same thing is going on. When the tumor becomes hypoxic, then hypoxia-inducible factor accumulates. But also there is this caveat, it also has pseudohypoxia. So how does this work? So in kidney cancer, 80% of patients, they have tumors with molecular program identical to one in hypoxia, even in the presence of oxygen. And here it is indicated as hypoxia-inducible factor plus group of patients. So how does that work? That molecular program is due to the loss of the tumor suppressor gene VHL. So let's just look at this one more time. So in the presence of oxygen, we have this modification of hypoxia-inducible factor. Then it is recognized by VHL tumor suppressor, and then this leads to distraction of hypoxia-inducible factor. So in the presence of oxygen, we do not have this transcription factor around. Now, what is going to happen if we lose VHL? Losing VHL, then in the presence of oxygen, hypoxia-inducible factor gets those modifications, but then there is no molecule which is going to recognize this molecule for distraction. And then there is no distraction of hypoxia-inducible factor. So oxygen or no oxygen, kidney cancer does not care. Kidney cancer always has high hypoxia-inducible factor. And then that is why it metastasizes so frequently. That is why this group of patients, which is indicated here as hypoxia-inducible factor positive, it survives less than the group of patients which is hypoxia-inducible factor negative. So kind of the same slides as this one, but then with the caveat that even in high oxygen tensions, those kidney cancers, they have high hypoxia-inducible factor and high metastasis. So another key term for today, cancer stem cells. This is something what was discovered 15 years ago, and then these are the cells which are among all cancerous cells in the tumor. There are a few which act as stem cells that produce themselves and sustain the cancer, much like normal stem cells introduced by you by Eileen. So if you look at the tumor now, it is not just a ball of cancer cells. It has cancer stem cells. It has cancer cells. We already talked about the blood vessels. They are right here. And there are a lot of different cell types which infiltrate the tumor. I am not going to touch those. And then what is the connection to hypoxia? I was very excited to see that several papers show that cancer stem cells prefer hypoxic microenvironment of the tumor to reside. So it makes sense. It is very difficult to target cancer stem cells with therapeutics. And this is because they reside in those hypoxic regions where it is difficult to deliver therapeutics because they are far away from the blood vessels. So I promised you to talk about two therapeutics we discovered here to treat kidney cancer. And then in order to introduce to you the therapeutics, I need to talk about synthetic lethality concept. First, I'm going to talk about normal cells, and then I'm going to talk about how we use the same concept in cancer cells. So in normal cells, we are going to talk about two hypothetical genes, gene A and gene B. So if we target gene A, then cells survive. If we target gene B, cells survive. But if we target both gene A and gene B, then cells die. So this is normal cells. So concept is called synthetic lethality. So cells die when there are two genes targeted at the same time. Now, how are we going to use it for cancer? So here we have kidney cancer. And then I just told you that this tumor suppressor gene, which is called VHL, the one which is distracting hypoxia-inducible factor, this is the one which is lost in 80% of all kidney cancers. 80% kidney cancers do not have VHL, so they have no gene A. But normal tissues, they all have gene A. Gene A is there. So this is a specific for kidney cancer. Now we need to find this magic gene B, which we are going to target. And then kidney cancer is going to die because it will have no gene A, no gene B, and then normal cells are going to survive because they will have no gene B and they will have gene A. So a very simple concept, but very powerful. We conducted a drug screen, and then we identified the drugs on multi-well plates, which are going to kill kidney cancer cells, and they are going to spare all of the cells which express the VHL tumor suppressor. So this is the example of an in vitro, so-called in vitro assay. So it is in tissue culture on the plate. When we plate the cells, we can use this drug. It is called Y27. It inhibits the kinase ROCK. And then right here, these are just 4 different cell lines showing the same thing. So I will concentrate on this part of the graph. So 100% corresponds to 100% survival of cells. And then 0 corresponds to 0 survival. So, and then here we have the drug concentration which increases. So as you can appreciate here, so this blue line, up to more than 10 micromolars of drug, cells are 100% viable. And then if you look at this orange line, these are the cancer cells which have no VHL gene. At this particular concentration, around 40% of cells survive. So there is a difference. There is a clear difference between the two, that normal cells which express VHL will be unaffected, but then cancer cells which lost VHL tumor suppressor will be affected. How does this look in the animals? So if we treat the animals with just the salt solution, where we usually dissolve the drug, so this is a no-drug treatment group, then the tumor steadily grows. But if we treat with the drug, then the tumor volume stays the same. These are the pictures. This is what we started from. This is the tumor volume. And then it stayed the same for the drug-treated group, and then it more than doubled for the group not treated with the drug. So we stabilized the tumor. It did not grow over the time. Another drug we also discovered through the same method, it is called fluvastatin. I've already mentioned statins are the anti-cholesterol drugs. They are used to lower the cholesterol. But also, they do other things as well. I will not go into details, but they also block a molecule which is very important for kidney cancer growth. So here we started from the tumor volume, 300 millimeters square. Then if we use the control, just the saline, then the tumor steadily grows. And if we use fluvastatin, administer fluvastatin to mice, then the tumor actually even regresses, the tumor volume drops. So these are the representative tumors. This is the control. And then these tumors are smaller. This is for 10 milligrams per kilogram, and this is for 15 milligrams per kilogram. They are even smaller. So this is all good. So now, before I go into summarizing this part of the talk, I will just go one more time through the key terms, and then I will talk also about the upcoming projects I just initiated at the Stem Cell Center. So today I introduced to you the blood vessels. So the blood vessels are the tubes in the body which deliver oxygen and nutrients to the tissues. I explained to you that every tissue requires a normal level of oxygen in order to function and generate energy. And when the oxygen is low, when there is hypoxia, this is when the tissue needs to adjust in order to survive and function. I talked about solid tumors and hypoxia. I told you that hypoxia arises because tumors are growing fast. And also their vasculature is not that functional. Sometimes it is prone to collapse, and then it blocks the oxygen flow. And then hypoxia is very important for tumor progression because it stimulates metastasis, and then patients die because of metastatic disease. Also, tumors turn on the formation of new blood vessels, and then the process is called angiogenesis. For the kidney cancer, I explained to you that they do have hypoxic regions as other solid tumors, but they also have a so-called pseudohypoxia. And this is when the oxygen tension is normal, but the cells think that they are in low oxygen. And then they have the hypoxia-inducible factor, the powerful transcription factor, which unleashes the program needed for survival in hypoxic regions and also drives metastasis. I told you about cancer stem cells, which prefer to reside in hypoxic regions. And then I introduced to you the synthetic lethality concept when we talked about gene A and gene B, and how in cancer we lose in 80% of kidney cancers the tumor suppressor gene VHL, which is our gene A, and then we are looking for the gene B to target for cancer cells to die. So I also told you about the ROCK inhibitors and statins as potential cancer therapeutics. And then the take-home messages from my work are that synthetic lethality approach is an important strategy for discovery of potential cancer therapeutics. Then kidney cancer with VHL tumor suppressor loss can be targeted by ROCK inhibitors and statins. As a future plan, of course, we want to confirm that cancer stem cells are VHL deficient, so they lost the VHL gene and then can be also targeted by ROCK inhibitors and statins. We do think that this is true because it is known that the VHL tumor suppressor is lost very early in the cancer evolution. So we think that the cancer stem cells also have VHL loss, but then this is something we are now looking into to make sure that this is the case. Also, we want to discover and test other drugs compatible with clinical trials for targeting kidney cancer. We have a drug in mind, which is also a ROCK inhibitor, and it is now in phase 1 clinical trials for tumors other than kidney cancer. So we want to repurpose it. And then we also want to test the ROCK inhibitors for targeting other types of cancer with hypoxic regions. So that's my last slide, the initiated cancer stem cell projects. So we do have the kidney cancer project where we are looking at the patient-derived xenografts and the 3D organoid cultures for drug discovery. And also we want to characterize those cancer stem cells a little bit better. And then also we have a breast cancer stem cell project. I'm collaborating with a very talented scientist here at UCI. And we want to know how cancer stem cells metastasize and how to target this. So this is the acknowledgement slide. So I would like to thank my lab and also my previous lab members and collaborators, and of course my funding sources. Thank you so much.
So I'm going to ask actually that we hold questions now because we're a little over time, and ask Dr. Fujitani to come up. so that we can make sure that everybody has a chance to hear the pieces that they're interested in tonight. So it's our pleasure to have here Roy Fujitani, who's a professor of clinical surgery here, and he's going to tell us about his research on the translational spectrum.
Thank you very much. Good evening. I do want to thank Professor Cummings and Anderson for the privilege of participating in this program. And I must say that this is very exciting, particularly because as I look out in the audience, I'm told that there are many young high school students out there. You can consider yourselves stem cells in the sense that you're undifferentiated and you've got a whole career ahead of you. And this is, just by your being here, shows that I think your career is going to be very bright. With that, I'd like to proceed. And what I'm going to change gears a little bit. I'm a clinician. I'm a vascular surgeon. And what that means is on a daily basis, I see patients and I operate on them, on their vascular system. And one of the areas that I will take care of patients is with those that might have advanced kidney disease. So just a disclaimer, one of the things I'm going to be talking about is a product called a human acellular vessel. And this is a conduit, that is a blood vessel that has not yet been approved by the FDA, but this is something that we have done in the translational spectrum. You had just heard about some of the basic science that goes into stem cell research. Well, this is the next step. This is where we've taken that science and now put it in the bedside and utilizing it in patients on a clinical basis. So the problem that I'd like to address, that's of kidney failure, end-stage renal disease. Many of you probably know someone, either a family member, a friend, or acquaintance who may have kidney disease. They may go on to have failure of their kidneys that they may end up needing some type of kidney replacement, either in the form of dialysis, peritoneal dialysis, or at best, kidney transplantation. But this is a worldwide problem. As you'll see from the slide here, the darkened areas are the dark red areas, that is, are the areas that have a high incidence, that is the diagnosis of new patients who have this disease. As it turns out, the countries that are most having most prevalence include Taiwan, the United States, there's Jalisco in Mexico, then followed by Thailand, Singapore, and North Korea, and finally Japan, which is very interesting. So this, in fact, shows the incidence rate. Now, one may be wondering, why do these particular countries, including the United States, may have a high incidence of renal failure? There is a direct correlation with the incidence of diabetes. Now, some of the other countries that you saw may not be as well represented, because if you don't count and if you don't record, well, you end up seemingly not having the disease. In actuality, these countries, including China, perhaps India, with very, very large populations, probably have as high, if not higher, incidence of end-stage renal disease. We're just not counting. And that's an important concept to understand. Now, when patients go on to have renal failure, as I mentioned, there are three modalities primarily in which kidneys are replaced. It was about 60 years ago that when a patient was diagnosed as having kidney failure, it was a death sentence. You got kidney failure, and pretty much there wasn't much hope beyond that. However, with the onset of hemodialysis, peritoneal dialysis, and kidney transplantation, that all changed. And you can see the distribution here that most patients today still receive hemodialysis. That is the exchange of blood through a kidney filtration machine that will then take over the function of the kidney. Okay? If the kidney does not work, your lifespan is not very long. It means almost certain death. In order to do that, however, clinicians such as myself, a surgeon, will not uncommonly have to provide a way in which blood can be taken out from the body be put into a dialysis machine, be filtered, and then be given back. In order to do that, one needs to have about 600 cc's, a little over a pint of blood every minute that is drawn out from the body, filtered, and then given back. Now, if you imagine, look at your veins in your arms, and if you put a needle in there, can you imagine drawing out a pint of blood every minute? Probably not. It's going to be very difficult to do. What we rely upon, however, are the veins and the associated arteries. In fact, if you just feel over onto your wrist and feel the end here, you'll feel your radial artery just on the thumb side of it. You'll feel a pulsation. Right next to that, you may or may not see a sizable vein. What we generally tend to do is we connect that vein and that artery together so that the vein, which normally has a low amount of flow, is now going to have a high amount of flow. And it's called an arterial venous fistula. That's what we do. So the National Kidney Foundation came up with a set of guidelines called initially it was called DOKI guidelines, which was the Dialysis Outcomes Quality Initiative. That came out in 1996. In doing so, however, they felt that, gosh, that's not very forthright in thinking about this. You really should think about the whole spectrum of patients having kidney disease. So they came up with the Kidney Disease Outcomes Quality Initiative. So that's why this is called the KDOKI, Kidney Disease Outcomes Quality Initiative, to improve the care of patients who have kidney disease. One of the initiatives to look at is how can we best serve patients by providing access so that they can receive hemodialysis. We try whenever possible creating what we'll call an autogenous fistula, using the patient's own artery and their own vein in their arms and connect them together and forming this access. However, the problem remains that not everyone has good veins. You may have heard people say, gosh, I got terrible veins, or my veins are very small. And in fact, that is true. We may not be able to do that. So if we don't have that, the second option then is to use a artificial conduit. And most often the conduit we use now is made out of a prosthetic material, most often Gore-Tex. The chemical term we use is expanded polytetrafluoroethylene, or EPTFE for short. It's a tube that's made from the PTFE, and we sew it into patients on both the arterial and venous ends, and that ends up being the way that patients get dialysis. On occasion, however, patients can have neither, or their disease progresses to a point that they need dialysis right away, where we don't have the opportunity to construct a fistula quite readily or quickly, in which case a catheter is placed. A catheter is often placed into a big vein, usually at the base of the neck or beneath the shoulders, but most often in the base of the neck, and that can serve as a temporary access. But of all the access that we can place in a patient, this is the least favorable. They get infected, they can cause blockages of the vein, and so we want to try to get those out as quickly as possible. So what does that mean? Well, what that means is that whenever possible, we try to avoid putting a catheter in. Okay? In fact, the rules are such that we should not leave a catheter in more than about 8 weeks. But that then ends up taking some foresight. The foresight is that we have to construct a fistula when we're anticipating a patient's going to probably end up needing hemodialysis. Okay? And so there are variations. We can do it in the forearm, we can do it in the upper arm, we can put grafts in there. But the point being is that there are many different ways in which to do this. The problem is when you create a fistula with your own artery and your own vein, they have to go through a process called maturation. That is, just because you connect the two vessels together, it doesn't get big or ready to use right away. And you end up having to spend some time for it to mature. It may take 6 weeks, 8 weeks, sometimes months before it is ready. All the while, they'll end up having a catheter, which is the culprit. We want to get rid of that. The other problem is that if you were to construct a graft and place it in, they tend to fail. And the reason why they tend to fail is that they form scar tissue, and it's called myointimal hyperplasia. It's a big word, just simply means that you develop scar tissue within it that can cause it to fail. And this is what it looks like. So this is a picture of a graft that is sewn onto the vein, and it's taking cross sections. So we section these, and you can see there's the lumen, the place where blood should normally flow, has become extremely narrowed, and that causes it to fail. And that, in fact, is the primary failure mode in which many of these access occurs. The graft that I just told you about, made of Gore-Tex, PTFE, it has nodes of tissue and fibrils between them that when you look under an electron microscope, through which tissue can grow. And that is an important concept. When we first started making artificial blood vessels of plastic, they're made of solid material. The problem is when you implant solid material into a patient, it doesn't allow tissue ingrowth. And that becomes very important. And they all ended up clotting off. They all thrombosed. The ability for tissue to grow into a blood vessel is important so that you can either have true or false, pseudo-false intimal ingrowth, meaning the growth of tissue within the blood vessel. The more of that intima, the inner line that you have, the more likely the graft will not fail. So that has led to the challenge that not all patients have good veins. Not all patients may be candidates to have this artificial graft, and if they have an artificial graft, that graft doesn't work as well. So this is where the translational research has come about, where biologic vascular grafts have been developed, and it's been a long time coming. It's well over 25 years in which many scientists have looked at this and determining whether or not this could be done. And in fact, for a while, it was being done, but these grafts took a long time to develop, and a patient needed this much sooner. And only more recently we've been able to develop a graft that can be synthesized. So what does that mean? Well, human vascular cells are cultured in a medium, and normally you can grow sheets of this on a culture culture plate. However, more recently, we are able to develop conduits, tubes of the same type of cultured cells that can be placed and made into a matrix. And it's made over a conduit, and it's put into a machine where over a 6-week period of time, these cells are then made into a tube, and that tube will oftentimes look like this. So this is cultured human cells. The issue is that if you take one cell line and then plant it into another patient, there's going to be a potential problem for angiogenesis and rejection. So what is done is that these conduits, these tubes that are then designed, are decellularized. So all the cells within the conduit are removed, and you end up having a collagen tube made of human tissue and the remnants of that. So when we look at the tube that is made from human cells into this conduit, you can see the normal cell structures, the normal types of collagen that you would see in blood cells. And ultimately, it comes out to a conduit that is made that can be now implantable, except this is a completely biologic conduit, biologic tube. And it has the same consistency as if we were to take a blood vessel out of yourself, such as the vein. Vein is a common blood vessel that we take out from the body and we transplant it to another part of the body to serve as an artery. Okay? And so there was experimentation initially done with this early on, up to about a year or so, and we found that this actually worked. And it worked well. It did not degenerate. There was no aneurysmal changes. That is, it didn't get big, it didn't burst. It tolerated arterial pressures. And so this is a picture of the conduit that has no cells before implantation. And after it's implanted into the animal models initially and more recently in humans, you can think of the conduit as being an empty condominium complex. And through that, the stem cells then invade into the progenitor cells, invade into this here once it's implanted, and those cells in turn will differentiate into blood vessel cells. Okay? Quite interesting, quite exciting. And in fact, the inner lining where blood is flowing, okay, this is the endothelial surface, turns out to become endothelial cells, just like normal blood cells. So in essence, we've fooled the body, if you will, to thinking that it's got its own blood vessel. The cells differentiate into the necessary cells that make it into a blood vessel. So what do we do? Well, we can now take this conduit, which is manufactured, takes 6 weeks, we refrigerate it, it's readily available. And we can then implant it into patients to become a conduit. So this is on the arterial side, that's onto the vein. We've constructed an arterial venous fistula. This is after it's healed, after about 4 weeks. This can now serve as a place where needles can be placed for purposes of hemodialysis, and even when a patient may not have a very good vein. And so that this is quite exciting. We've initiated this as one of the clinical sites in which this is implemented. So what's exciting about this is that we call it the human acellular vessel. The vessel gets populated with progenitor cells, and over time, and up to like over a year, what you end up seeing is that the conduit, the artificial biologic conduit that was placed in there, looks very similar and very much like a normal human blood vessel. Okay? And because these conduits need to be needled 3 times a week, they have a lot of injury to them. And because they have injury, one of the concerns has been, well, where you injure these blood vessels to place the needle in, will they heal? Will they heal normally? Will they become aneurysmal? Will they become large? And as it turns out, they do heal. And they heal very similar to that of normal blood vessels. That's an important concept. And so it repopulates even after the vessel is in fact injured. So when we looked at this in the first-in-man type of results, what we saw is, for one thing, when we looked at for immunogenicity, that is, did this cause an immune response to patients when there was implanted? And it turned out no. They did not develop an immune response. And that's very important, because in other words, this is not like a transplanted tissue, although this came originally from another human in which this cellular line had been made. But because it had been decellularized, there does not appear to have caused an immune response. In addition, there was no aneurysm formation, and the flow rates, that becomes very important. Normally, these things have to flow at about 600 cc's a minute. It could tolerate that very well, like a normal blood vessel would. Additionally, patency. Now, what does that mean? That means that it has to stay open. One of the biggest problems we have as surgeons is that when a patient has a fistula constructed, they go on to fail, much because of the scar tissue that I showed you earlier that can form in the blood vessels. So will this mean that there would be a higher incidence of scar tissue or so forth? As it turned out, no. In fact, the patency, the ability to stay open, has been superior to that of prosthetic grafts, those made out of Gore-Tex or polytetrafluoroethylene. And that has been quite exciting as well. So to summarize, I'd like to say as an audience, what we're excited about with this whole stem cell therapy with regard to vascular surgery and our ability to treat patients with end-stage renal disease is we now have, it appears, a first off-the-shelf bioengineered vascular tissue that we can implant into patients. It's non-immunogenic. It integrates very well with native tissue on the arterial end, the venous end. It can repopulate, it can remodel as necessary, because that's what happens to these constantly traumatized blood vessels. Looks like it maintains the strength and has little intimal hyperplasia, the scar tissue that forms inside of there. We are participating in the phase 2 clinical trials with this, and we're proud to say that we're the second leading enroller for these clinical trials with regard to this. We're also initiating this very same technology in trauma patients. So that imagine now if someone comes in with a traumatic injury to their limb and they have lost blood supply to their limb, and we now need to resupply blood to that limb, and they may not have a very good conduit. They may not have a vein. We can potentially take this off the shelf and use this as a bypass graft in place of a plastic or prosthetic graft to be used in those patients. So with that, I'll end. And again, I greatly appreciate the opportunity to present to this exciting work with you. And again, I thank you very much for your kind attention.
So we have plenty of time for questions. Thank you very much. I think those were really a pair of great, very clear presentations. I hope you guys came away with the idea that stem cells really mean a lot of different things. I think that's one thing that was great here, right? From the idea of stem cells in development to stem cells in disease, like cancer, and what a causative agent they can be, to how we can use them in really maybe unexpected ways to think about repair, even making biomaterials, right? Using those cells to make things that we need for transplants. So anybody who has any questions, I think every student here is required to ask at least one. Please ask questions. I'll bring a microphone around. I think we have an extra one so that you can hear.
Something I want to tell to our high school students. Keep in mind, you know, I'm thinking back when I was a high school student where you are now. I don't think I've had that opportunity way back when to do what you're doing. And so ask a question. You know why? Because if you're thinking about it, I'm sure that someone else in the audience is thinking that same question. The only foolish question will be the one that's unasked. And you're going to go home a lot smarter if you ask that question. We may not have the answers, but if we do, we'll answer those.
If you kind of yell, then we can repeat it.
I just need a little bit of clarification on the GA and GD part, because on my note, because it was in the back and maybe it wasn't that clear. So you said that if you only have GA or GD, you will be a normal person, but if you have both, then you will get cancer, right? I was confused on that part.
Yes, so the question was to repeat what the gene A and gene B meant for the synthetic lethality concept. So I will go into that just in a few words. So these are hypothetical genes, and we are talking about the level of the cells. So we are not talking about the organism. Yes, so at the level of the organism, if we just extract cells from the organism, normal cells, and then we affect gene A, then cells are going to survive. Or we affect gene B, cells are going to survive, but if we affect both, then they are going to die. So, and then in cancer, gene A is already absent. The VHL gene is already lost. That's a tumor suppressor, it is mutated. And in normal cells, it is present. So we have a difference now between cancer cells and normal cells. And then we need to find something which is going to target gene B in order to kill cancer cells, because then they will have gene A and gene B affected. But then normal cells will still have gene A, so they are going to be fine. This is just a hypothetical thing. For every single cancer, it is going to be different. So for kidney cancer, our gene A is VHL because it is lost in 80% of cancers. Of kidney cancers. Then let's say for some other cancer it is going to to be a p53 gene. For some other type of cancer, it is going to be a RAS oncogene gene. So there are, depending on the cancer, we can pick a gene.
So
Yes.
because I'm not a cancer biologist, I'm going to say that back to you in a slightly different way. So lots of genes. Genes all make proteins, right? They're really important for what our function is, but they are redundant. So one gene can pick up the slack for a different one to a certain extent. So you can lose one and be okay. But when you lose that certain one that's picking up the slack, that's when the cell dies. And so gene A, gene B could be anything where two genes are working together for survival. Lose one and you're all right, but what Olga's trying to do is target that other one that's normally picking up the slack and then get rid of it as a way to target cancer cells. Does that make sense?
I was just going to ask, like, in the cancer cell, so if there's a gene A and gene B, and without the gene A, it doesn't have VHL. So when you're using that, you have to target the A cell. You have to kill the B cell, or target the B cell, so that way there's both of the cells are gone, and that's how you get rid of it.
So it is inside the same cell. So cancer cells, they have a mutation in VHL, so the gene A is already absent. And then we need to target another gene in the same cancer cell in order to kill them. And then, as Eileen just said, so there are parallel processes, and then they are operated by parallel so-called signaling pathways. So VHL is controlling one signaling pathway, and then there is another one, which is doing something similar. And if we disrupt both, then it is incompatible with cell viability, so cells are going to die. Yes.
How do you sew those vessels to another vessel?
So the question is, how do you sew one vessel to another vessel?
Were you really good at home ec when you were...
It's the essence of being a surgeon. Yeah, it's well, actually, as it turns out, these blood vessels, artificial blood vessels, biologic blood vessels, handle very similar to that of our normal blood vessels. In other words, if you go to a surgeon and let's say your blood supply to your leg is not very good, so one option is to open up the blood vessels that are there, or you can take your vein, which are normally blood vessels that return blood to the heart, and you can sew them onto the artery, and it in turn becomes the artery. With these artificial blood vessels, we sew it in with sutures, most often with a polypropylene suture, nylon. It's like fishing line, and various degrees of strength on both the arterial and the venous end, if we're doing a fistula, or artery to artery if we're doing an artery.
Yes?
In regards to the artificial vessel, you said you depopulate the cells that were in there and then they insert it, and then the new stem cells integrate themselves into that vessel and start to grow new tissue. I was unclear on if the body is actually able to populate it with stem cells, or are you injecting stem cells into that area?
What's amazing about this is that this conduit is then tunneled in the patient, usually in the subcutaneous space, that is underneath the skin, between the muscle and the fatty layer. And a lot of the stem cells, progenitor cells that reside in those tissues, then go on to populate that conduit that's there. So none of this is done beforehand. This is something that happens naturally once the conduit is in place.
Could you clarify pseudo-hypoxia? Because does that mean that like the cells don't react based on like the amount of oxygen?
This is exactly right. So the question is to clarify the pseudo-hypoxia. So basically, in hypoxia, cells sense that the oxygen concentration is low, and they sense by accumulation and stabilization of this hypoxia-inducible factor. This is the key molecule to maintain cells alive in hypoxia. But then pseudohypoxia, this is when cells don't know if there is a lot of oxygen or there is little oxygen. They have this hypoxia-inducible factor in place. So if they have low oxygen, it makes sense. This is how cells respond to low oxygen. But if there is high oxygen, normal cells, they have no hypoxia-inducible factor there. But then kidney cancer cells, they still have a lot of that, independent on the oxygen concentration. So that is why it's pseudohypoxia. It is cells are responding the same way as they would respond to low oxygen, but they don't sense oxygen. Okay. Yes?
Is there a way to treat hypoxia, or is it?
That's an excellent question. The question is if there is a way to treat hypoxia. So those attempts were actually done, and yes? Yes, give me just one moment. So basically cells, so researchers have figured out that we can actually use the vasodilators, and then this is what increases the oxygenation. Then also we can use some kind of hypoxia mimetics as well. So this is something what is now used in the clinic in order to oxygenate the tumors better, but at the same time the outcome is not that great. So we cannot oxygenate them to the level which is required. Also very counterintuitive. We actually have a lot of therapeutics which kill the blood vessels. They're called anti-angiogenic therapies. And then they actually do help because you kill the blood vessels, then tumor cells cannot get oxygen, cannot get nutrients, they stop growing. But the downside of that, they are going to metastasize more effectively. So still they are used in the clinic a lot, and they are helping. They are prolonging survival like by 6 months for the patients. They are better than nothing. But then unfortunately patients do relapse with metastasis because of that.
Yes, I'm here for my mom. She have a 2 kidney failure, and she on dialysis for 4 times a week. And you think that stem cell can help her in the future to heal her better, can treat her?
So first of all, I'm sorry to hear about your mom, but on the other hand, it sounds like she's successfully being treated 4 times a week, which is good. So that, in essence, implies that she has adequate access in order to be treated. Now, the question then, and we can talk about this later, perhaps privately, but it depends on the kind of conduit that's there. If she ever gets to a point where these fistulae stop working or there are no other options, then the answer is yes. There could be opportunity to help her, but it depends. The point being is that when patients have kidney failure and they end up needing to be on dialysis, that access, their arterial venous fistula or graft, becomes their lifeline. And the problem is that on average, those don't last. They can go on to fail. And after a period of time, if they continue to fail, you run out of options. And this is where this might be helpful. Okay.
So if I may, I'll just interject really quickly, maybe away from that speaker. I think you're maybe angling at a slightly different question as well, which is about can you replace a kidney with stem cells? And do you want to take that, or do you want?
Well, that would be outside of my area of expertise.
So I thought I would just comment quickly, and I'd be glad to talk to you. Not my area of expertise either, other than the idea of what's called organ and tissue engineering, which is something that's really important and a big part of stem cell research right now. So in fact, we just as a center started a collaboration with a university in the Netherlands, University of Maastricht, which has a program to build a new kidney. And their idea is to use 3-dimensional organ and tissue engineering with stem cells to be able to make replacement organs in the long run. There's a lot of interest in this sort of thing for different sorts of organs and tissue. And I think it's the next generation of stem cell research that is just really getting off the ground in that pipeline, right? At the very basic science level, moving on up through those stages we talked about. I saw there was, yep.
Can you access a patient if they have, like, I guess, if the veins maybe aren't getting enough flow, but they're still able to be accessed? Can you do it in two different places?
As a matter of fact, the question is, can you access these conduits, you're saying, in two different places? As a matter of fact, when patients do get dialysis, there has to be an afferent and efferent limb. What that means, there need to be somewhere to get blood from and another one to give it back. So all of these access almost invariably have two places that they get stuck. It's a pretty big needle. It's like a 14 gauge needle, which means it's a big needle. And so, and doing so, they need to do that because, as I mentioned, you need to get it anywhere between 400 to 600 cc's every minute. And just imagine if your vein were being stuck in two places 3 times a week, sometimes 4 times a week, what kind of abuse that will be taking. And that's what happens to these conduits. So I hope I answered that question. Yes?
What's the materials that you use for the conduits again?
So the primary conduit, the question is what other materials are being used. So the primary conduit we use aside from polytetrafluoroethylene, expanded polytetrafluoroethylene. Gore-Tex is another name for it, but there's several manufacturers that do that. We do that for bypasses of arteries. We do that for arterial venous fistulae. There are other materials include xenografts. Xenografts simply mean that these are blood vessels from other species, cow carotid arteries, cow mesenteric veins are ones that we will sometimes use as conduits. They tend not to work as well, and they tend to cause sometimes some antigenic problems. There. Any question back there? Yeah, I'm sorry, we can't hear you, so maybe we'll get the microphone back there.
So can you replace every kind of vessel or just specific kinds of vessel?
So keeping in mind this is very early in our clinical trials with regard to, we're specifically looking at this in patients with renal failure and doing what we call arterial venous fistula. So we're connected to the artery, connected to the vein. We are going to go into clinical trials. We're going to connect an artery to an artery. But this becomes a conduit. To answer your question, ultimately, the goal would be to have these types of vessels be usable in just about any blood vessel. And because these are constructed over mandrels, that is molds, it's quite possible in the future we might be able to do tapered grafts, branch grafts that can mimic that of normal anatomy that might be used to replace blood vessels. So that's still yet to come, but I anticipate that should be coming in the near future. Yes, Dr. Galb.
I think this is enormously exciting, but I want to ask about a disaster in Europe with artificial trachea, where they attempted to build new trachea from plastics seeded with stem cells, and they never remained patent with disastrous clinical consequences. Why do these stay open so effectively?
I don't have the scientific answer for that. We know that these are high flow conduits. They tend to be bigger conduits, at least 6 millimeters in diameter. And so far from the preclinical and clinical trials, they appear to do so. Mostly, vessels tend to stay open if you can get a very good intimal lining. In essence, I think for those that may know, this is the cell lining inside a blood vessel. It itself is considered an organ. So that inside of all of our blood vessels, there's that inner lining called the intima, and that intima itself it's an anticoagulant. It keeps vessels open, keeps it from clotting, And it appears, at least, that by mimicking that of a true blood vessel, it tends to stay open.
One last question.
Might be silly, but is that Gore-Tex that you use, is that the same Gore-Tex that's used in clothing and shoes that?
That's the one.
Yeah, but they're kind of a plain vanilla white colored material. Yeah, but it's expanded polytetrafluoroethylene. And so let me explain that to you just a little bit. It simply means that DuPont, a number of years ago, had come up with this powder for polytetrafluoroethylene. Didn't know what to do with it. So send it off to clinicians, say, you might want to think about using this. So they, in fact, looked at that. They took this powder and expanded it. So that means there's nodes of tissue with fibrils that go in between that most of the graft, as it turns out, if you look under a microscope, 80% of it's air. With materials that you use, like your Gore-Tex shoes and jackets, and that's made into more like a textile. And so it's sewn and everything like woven or knitted to make what it is. So it's a little different, but it's the same material that's used.
So I just want to thank everyone for coming tonight. I hope you enjoyed the lectures. I did. Thank our speakers one last time.